Okay, so let's try this. I've managed to work out a method of balancing my phone on the visualizer so that I can explain what's going on in the cardiac cycle. Many of you were in for this lesson, but some weren't, and I think it's worthwhile going over what's happening. What's not in your notes is the electronic control of the cardiac cycle that we do later on, but I think it's worthwhile knowing what's happening as we go through looking at the actual cardiac cycle. So we'll start with the cardiac cycle. If you look at the diagrams of the heart, you can see that we've got them in 3 phases. In different diagrams, you'll have different number of hearts all doing different things. If you've had a go at the flick book, then hopefully you've got used to the idea of what's happening in each different stage. At the different stages, you should be looking at the volume inside the areas, and you should be thinking about what's happening to the pressure, whether the chamber is relaxing, as with these, or contracting, as with these. When they're contracting, they're being indicated by little arrows, and the contraction, if you remember from the previous lesson, is that's when we call it systole. And when they are relaxing, like these, they're in a diastole. Now, because it's a cycle, you can start in any area you like, but I like to start in this one, because in this one you have got complete diastole, and I recommend putting little d's on there like that. I remember that the diastole is the relaxing one, because diastole means, well, to me, D for dead. It's not actually dead, but it's relaxing. So here in diastole, what is happening is that all 4 chambers are relaxed, and the blood that's pumping around the body is now coming into the atria at the top. What happens in one side happens in the other. So if I just mention it on one side, I'm automatically assuming you realise it's happening on the other side as well. So the blood, after it's been pumped round to the lungs through this one, or the rest of the body through the aorta, it flows back in again. It's flowing back in passively. And as it's flowing back in passively, you get what they term passive filling in the atria. Now, the passive filling in the atria allows the blood to come in, but as you can see, all the valves are closed at this point. The valves are closed because the pressure is not big enough to open the valves. So the blood is flowing into here, into the atria. The blood is going in and in and in. As it's coming in, the pressure is starting to increase. Now, hopefully, you've already done the notes and you've looked at the pressure curves, and you'll be thinking, right, that's why the pressure is going up in the atria during atrial diastole. So the blood is going in, the pressure is increasing, and when the pressure gets above that of the ventricle, the AV valves open. These are the atrioventricular valves, AV valves. The AV valves open, and the blood then passively fills the ventricle. So the blood then moves down here into the ventricle. But that's when we move on to the next one. So at this point in time, we've got passive filling of the atria, and the AV valves are shut. You can also see that the semilunar valves, which is the pulmonary valve, and the aortic valves are also shut. So we go from here back to the beginning, and in here you've again got passive filling of the ventricles. So the blood is going from here into there. Now, because the blood is going from here into there and the valves are open, then you have not got isovolumetric filling. Over here, you've got isovolumetric relaxation. What that means is that the volume in all of these chambers remains the same. The pressure will be increasing in here until it's above that, but it stays the same volume, isovolumetric. So here, the blood then flows down from the atria into the ventricles. This is passive, so you've got passive filling. So this is passive filling into the ventricles. As you see, the AV valves are open, the semilunar valves are still shut, and here this is in diastole, and this again is in diastole. Then we go to atrial contraction. So here we've got atrial systole, we've got ventricular diastole. Now, the last 10% of blood that goes from the atria into the ventricles, so that's just the last 10%, gets pumped through there. I'd like you to know that it's just the last 10% that goes through there. So the last 10% goes through into the ventricle. And this is done when the atria is in systole, the ventricles in diastole. Then after this stage, we go into ventricular systole. Atria then go into diastole, so the atria then relax. And this is then known as isovolumetric contraction, because as the ventricle is contracting, the volume is decreasing. Sorry, not the volume, the pressure is increasing. So it's squeezing it, the pressure is increasing. You've still got the same volume inside there, even though it's squeezing, the same volume of blood is inside there, inside the ventricle, until the pressure inside the ventricle is higher than that of the atria. When the pressure here is higher than there, the AV valves are shut, as you can see in this picture here. So when the pressure in here is higher than there, those shut. But the pressure at the moment is not high enough to open the semilunar valve. So this is isovolumetric. It's isovolumetric because the volume of blood in here stays the same. Then, once the pressure, when you've got atrial systole, ventricular systole, atrial diastole, the ventricle is contracting to such an extent that the pressure in the ventricle is higher than the aorta and the pulmonary artery, and therefore the semilunar valves open, allowing the blood to move up and out to the rest of the body, to the lungs or the rest of the body. So that is then ventricular ejection phase. So you've got the ventricles in systole, allowing the blood to go up and out. Now what goes up must then come back down again. The blood comes back down again because the ventricle goes into diastole, and when the ventricle goes into diastole, rather than squeezing the blood upwards like that, it then goes into diastole. When it goes into diastole, the volume increases and the pressure reduces above that of the arteries, and because the pressure is so much lower than that of the arteries, the blood then gets sucked back like that. The blood then gets sucked back, forcing the semilunar valves to close. When the semilunar valves close, that causes a noise to occur, which is the lub-dub sound. So lub-dub, 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 when you listen to your heart through a stethoscope. Now, that's the base of what you need to know at the moment, but you'll also need to know how that is controlled. And in one of the classes, I did go through that, but we were a bit of a rush because we knew we were closing, so I didn't go through it with the other one. But I think it's important that I cover the same thing with everybody. Now, we've got a diagram, basic diagram of a heart here. You all know the different parts of the heart. I'm not going to be labelling them on there. The thing that we're going to be looking at is what controls the heart rate, the pulse of the heart. The thing that controls the pulse of the heart is the SAN, sinoatrial node. The sinoatrial node, otherwise called the pacemaker. Pacemaker. So SAN, sinoatrial node. So sinoatrial node, otherwise known as the pacemaker, is located in the wall of the right atrium, like that. We also need to have the atrioventricular node, which is again in the atria, but more towards the septum, sort of like around there. And that is the atrioventricular node, or AVN. So you've got the AVN there. Now, I'm only going to cover this sort of like briefly. We'll do a lot more on it when we do the topic on this. So you've got your SAN, which is that one there. Then you've got your AVN, which is this one here. Now, these two structures are part muscle, part nervous. You've also got things that go, so it's not the best of diagrams, I could have downloaded a better one. Down the septum you have fibres that go up and along. Now these have a strange name that can be called two different things. I call them the Purkinje fibres, or they can be called the Purkinje fibres. So these Purkinje fibres, as you can see, I've sort of like done a sketchy type thing because they are they are sort of like fibres. The part muscle, part nerve. And because the heart itself is myogenic, I did mention that term to you before. Myogenic means that they've got the ability to contract without external stimulus. The Purkinje fibres, there are lots of fibres going down the septum, which is that middle bit there. So on the diagram up here, it's there. So the septum, down the septum, and then up the sides. You've got your Purkinje fibres. And collectively, they're called, very strange name, bundles of His. I think it's a person called His, and that's what there's bundles of fibres, and they're named after the man. So we've got that bit. Next, so we've got the SAN up in the right atria. Then we've got the AVN that's in the septum. You've got your Purkinje fibres or bundles of His that radiate down the septum and then back up again. Then we need to know one more thing about the sort of like anatomy of the heart, is that between the call that in like that, between the atrium ventricle, you have non-conductive tissue. And this non-conductive tissue prevents any electrical stimulation going downwards. So what actually happens is that you have electrical stimulation radiating out from the SAN along and throughout both atria at the same time. Now, I think if you were in the lesson, but I'll mention it again in case you weren't, muscle tissue. Now, you've got 3 different types of muscle tissue. This is sort of like a very simple diagram of skeletal muscle. Skeletal muscle is the type of muscle that you would contract, sort of like move your bones, so lifting your arm. So anything that you've got control over is skeletal muscle. And yes, you're right, I've got more than one nuclei in there because it's multinucleate, but you'll learn more about that in year 13. So that's skeletal muscle. Smooth muscle, I'll put one on there. Smooth muscle looks a bit more like that. We'll make it into a skeletal. Smooth muscle is it will have a nucleus, but it's not got the same striations. The striations are made from the muscle fibres we talk about next year. And that's the type of muscle you don't have control over, which are things like your esophagus and peristalsis, and the way in which your pupils can get bigger and smaller, dilate and constrict. But we'll turn it into a striated muscle. Now, cardiac muscle, which is what the heart's made from, is slightly different in structure. It's got cytoplasmic interconnections like this. And these cytoplasmic interconnections make it a lot easier for the electrical stimulus to move along the membrane and to the next cell. So it makes the transmission of electrical impulse much, much faster. We'll do next year in year 13 about how impulses actually travel along a membrane. It's quite a detail. It's exciting. You'll enjoy it. Right, so back to this. So you've got your SAN sends out a wave of excitation across the atria. That wave of excitation, the wave of electricity, causes the atria contract. So you've got atrial systole. Now, once you've got atrial systole, which is happening here, the electrical impulse comes along, causes the atria contract, but then the impulse can't get any further than here because of the non-conductive tissue. Hello? I'm just making a video. The non-conductive tissue. So, And we don't want the impulse to keep going downwards. If the impulse kept going downwards, then you'd have a contraction of the atria going right down to the bottom, and then the blood would have to come out the bottom, which doesn't happen. As you all know, it comes out through the pulmonary artery and the aorta. So the impulse, because it can't go down because the non-conductive tissue is picked up by the AVN. And from the AVN, the impulse then goes down the Purkinje fibres and then radiates upwards. So the impulse then comes down and then radiates upwards. Now there's a small delay. Now the examiners like you to mention this delay. There's a small delay in the impulse once it reaches the AVN before it then comes down again. And the reason why it's important that there's a small delay here is so that if we go back up to this diagram, is so that the atria can fully empty into the ventricle. If the ventricle started in systole before the atria had emptied, then you wouldn't have efficient blood flow around the heart. So the impulse then goes down before it radiates and comes back up again. So just in summary, so as you can see, it does all really fit together. So you've got your SAN up here, you've got your AVN here, you've got your non-conductive tissue that goes across, you've got your Purkinje fibres and your bundles of His that go up. Your Purkinje fibres collectively are called the bundles of His, and they are cross between muscle and nervous tissue. They carry the electrical stimulation down and then radiate up. So your impulse is a wave of excitation across the atria, causing the atria to contract, going in atrial systole like this diagram here. Then they prevent it from going any further down because of the non-conductive tissue. The impulse is picked up by the AVN, which is in the septum. The impulse, then there's a slight delay to allow emptying of the atria. Then the impulse radiates down towards the apex of the heart, and then the impulses then move upwards. If you've got any questions, please email.